So we will start. I don't think I'll do a big introduction because this morning was so interested and lasted a bit more than 20 minutes more than expected. So I just go straight on to our next speakers. Just to remind you that this now we will focus on the indicators, soil biodiversity indicators, something that in this morning the Agri kind of focused on. And we will start with Julia. Julie Bongiorno, yes, and also our colleague that we already had the opportunity to listen to this morning. Both of them researchers. We start with Julia from Wageningen University, from the soil microbiology biology group. Well, and without further ado.
Yes. Okay, great. Thanks, Anna. Thank everybody for being here. Okay, so a little, how does this work? Yeah, okay, a little outline. So I'm gonna talk and give a little presentation introduction about biodiversity monitoring methodology. So we start from general, and then we will go a bit more in depth into the methodologies that we use in SOILGUARD. So I will give a bit of a more background to actually the results that Pablo presented previously. Then I will give a little insights into some preliminary results regarding comparison between different methodologies, molecular versus microscopic, for some particularly soil fauna groups that we characterize in SOILGUARD. And then I will give back the words to Pablo, and he will present the soil biodiversity indicator that came out from the SOILGUARD project. So first of all, the EU directive on soil monitoring and resilience, since we all know, is quite important in the framework of the EU soil strategy for 2030 and consequently also for the European Green Deal and the EU biodiversity strategy. But within this monitoring and resilience directive, with this directive, one of the key soil threats that is addressed is the loss of biodiversity, among other threats that are really, impairing and having a damage on soil. Why? Well, because soil biodiversity is very important. There is an intrinsic value of soil biodiversity, but different soil biota are very important for a variety of functions that we don't have time to go in depth here, but they are really underlining and very important for very important processes in soil. So we have a huge variety of soil biota living in soils, and there are various measurements, various indicators that we usually assess when we measure soil biodiversities are mainly biomass and abundance, depending on the indicators of the organism, the richness, so how many species or specific taxonomic group are present of specific soil organism, and another one is also very important, the community composition, and we refer to these indicators before as alpha and beta diversity, if you remember previous presentations. But the big questions, of course, when you start actually measuring this organism is how. And here we are gonna go a bit into more depth into different methodologies that we have available to measure soil biodiversity. So I'm gonna present three main methods. So the first one, is a morphological method. So for various organisms, in particular, the fauna, so the bigger organism, microfauna, mesofauna, macrofauna, we have available morphological methods. So we extract organisms from the soil, and then under the microscope we have the capability to count them, how many they are, but also who's there. And we do this, usually for soil fauna, for example, you can see here, nematodes, earthworms. Maybe not all of you are familiar with this organism, but we are quite a lot. So these are the methods that are mainly used for fauna. For mainly soil microorganisms, we can assess certain particular components, chemical components of these microorganisms. For example, PLFA really targets components of the membranes of this organism that we can see here in this depiction, and this is usually used for soil microorganism. But we also have another method that in these last years been used a lot, usually is used for soil microorganisms because it really targets also the part of microorganisms that we cannot culture, which is quite a lot. Molecular methods really targeting DNA and RNA information present in the soil. In recent years, these molecular methods have been also applied for fauna, which is, let's say, well, it's a more advancement of these methods, because usually, as I mentioned before, fauna is assessed through morphological method, which is considered the golden standard. And so the method that we use in SOILGUARD is eDNA. So basically, we have here our soils, we have here organisms that we can see, but we also have organisms that we cannot see, like microorganisms. And from soil directly from the soil, so not extracting organism before, we can have DNA, environmental DNA, which can then be characterized through, for example, sequencing, so having a more qualitative idea of the communities, or qPCR, or more quantitative informations, and then we can actually characterize a huge variety of organisms, microorganisms, but also fauna. And in these samples, we can have information about abundance, richness, community composition. And this is what was actually done in the context of the SOILGUARD project, which this abundance, richness, and community composition, for the moment, are the variables that were mainly analyzed. And you can understand that with this method, there is a high, well, they're highly beneficial, and they have an important role, because they could be used to, be more through output, so they could be used at a bigger scales of characterization when we sample. They could be very useful in terms of harmonizations of the methodology for measuring soil biodiversity, because the same methods could be applied everywhere, and could also facilitate the identification of these organisms. But of course, this only if the taxonomic information that are available in the database that really gives an indication of, okay, to who this DNA belongs to, if those databases are good enough and are complete enough, which is not always the case for all the organisms that we want to characterize in soils. So for SOILGUARD, we use 3 different methods. We use all of them depending on the indicator. So we use more golden standard morphological methods for nematodes, acari, and collembola, which are micro and mesofauna. We use the PLFA, so this characterization of the quality of the membranes, for bacteria, fungi, and protists. And we also use the eDNA methods for a huge variety of organisms, including bacteria, fungi, viruses, protists, nematodes, acari, collembola, and earthworms. So what we see here is that basically we use different methods for different organisms, and that is when comparison between different methods come at a useful point, right? Because we have different methods available for the same organisms. The new developments, as I already mentioned, have big potential, so they can be very useful, but are they reliable? So these new methods, are they reliable? How do they compare? How do they how do they compare with the traditional golden methods? So what I'm doing in these months, and still an ongoing project, but I will show you some preliminary results, is the comparison of this 3 group of organisms, nematodes, acari, and collembola. So I'm trying to understand how the results from the molecular compare to the results for the morphological, to understand if these new molecular methods, methodologies can be used, and how much they are useful for us. So here I will go a bit into the results. I will try to be brief because I don't have a lot of time. Here I will show you the similarity index, which basically is linked to the families, to the presence, absence of families, in this case of nematodes, and basically it shows for each sample how much similar is depending on the methods used. So how much each sample, the 2 methods are agreeing in finding this taxa. So for the nematodes, well, we already see that actually in terms of number of family, we found 57 family for the molecular and 47 for the morphological. So actually that's not so much difference in terms of number of family that we found in the samples. But when we look at the similarity index, maybe you don't see very much here, but we have in this graph, we have the frequency, so the percentage of samples that have a certain level of similarity. So the higher is the similarity index, the more the 2 different results from the 2 different methods will be similar. And we see here that for nematodes, the highest number, the highest percentage of samples have a similarity index between the two different methods around 50 to 60%. So, well, it seems not so bad, but it also depends from which side of the coin you look at it. But when we look at Acari and Collembola, which is a mesofauna group, microarthropods, well, the number of families that we found with the molecular was 34 versus 74 families found with the morphological. So already there we see quite a discrepancy within the two methods, and the similarity index was actually much lower, with quite a lot of sample even not being similar at all, and the highest percentage of sample between 11 to 20% similarity between the two methods. So we can already see that for some organism, the two methods compare quite differently. So now I will go a bit more in depth into the nematodes analysis, because this is the more advanced one. And I want to show here the percentage of shared family between the two different methods. We found around, 57.6% of families, of all the families that we found were shared between the two different methods, and only 13.6% with only found with morphological, and 28.8% with the molecular. And this is probably can be explained by different reasons. For example, here in the morphological ones, the amounts of these organisms found were so low that they might not be picked up with molecular methods, or maybe there are some issues with the molecular databases. So maybe the databases is not developed enough in order to give a good identification. And then here, among the samples, among the taxa that we found only in the molecular, there are various reasons. Maybe I should not go too much in depth into this, but for example, there are definitely some taxonomic issues, some extraction issues. Some are never found actually in the microscopic, or also there might be the problem that actually some relic DNAs, there's some DNA from nematodes that were there years ago and that stay stable in the soil, but that are not really corresponding to nematodes that are living in the moment in the soil, which are actually the ones that you found with the morphological ones. And then when we go to a bit more into looking in functionality, so this is the maturity index is an ecological measure of environmental disturbance, which is based on nematode species composition. So you can have idea about more the functionality of the system because of the characteristic of nematodes that are present in the soil. We can see that, and this index is very used as a nematode-based soil health indicators. It's quite useful. We can see actually that the correlation between the maturity index with molecular versus microscopic methods is quite poor. So this also kind of brings us question about, okay, but if you use this molecular method to drive more functional characteristic or soil health characteristic of soil, can we really rely on these methods? So some take-home messages, we see that different methods are available depending on the organisms. And we also see that new methodologies are available, but it's very important to compare them with golden standard. And because what we also found is that there is little agreement between morphological and molecular methods for soil fauna, in particular for Acari and Collembola. And of course, this brings us to really think that each method has its own limitations, some limitations for the morphological, some for the molecular. But the question is, could maybe these different methods be used complementary in the end? And could we maybe actually look into more, ways in which we can make actually better molecular identification? For example, in case of soil fauna, extract first soil fauna and then characterize the DNA without using eDNA. But this is, again, a question. So basically, I plea for a careful interpretation of results from novel methods, because this is what we found. And I think there will be time later for questions. Now I want to invite Pablo to continue with telling us a bit about indicators.
Okay, hi everybody again. I will continue this analysis of the soil biodiversity of SOILGUARD, in this case focusing on all the metrics that Julia explained before related with richness, diversity and abundance of the soil organism in the soil. And for this, we tried to understand how much information we can obtain from the soil ecosystem with a fewer numbers of soil biodiversity descriptors, or also called bioindicators. First of all, we want to say, to acknowledge how complex is the soil interactions that we observe, and this complexity is really needed to understand what is happening in terms of the soil, of the health of the soil. As has been previously discussed in the soil community, and more recently some initiatives, as the ones from SOILBOND, made an effort to outline several frameworks where we can understand how many different interactions can happen in the soil, and what are some of the indicators that can give us information about these interactions. However, this complexity, and maybe also the lack of awareness in the past of the soil life, has led to a quite imbalanced presence of soil biodiversity metrics in soil quality assessment, as has been described by this review, where they compile a lot of soil quality assessment, where most of them has typical soil properties well known as the total organic matter and pH in almost all the studies. It's close to the 90%. And in terms of the green ones that belong to the biodiversity, these are below the 40% of the data sets related to soil quality assessment. So in this line, what we need, as it was mentioned at the beginning of these sessions, of the previous session, is that we need something robust, feasible, easy to interpret, harmonized, and also with, let's say, a feasible number of biodiversity metrics that can help to, let's say, to establish future global monitoring in a successful way. In view of that, we have these main objectives, was to establish a minimum set of indicators that help us to assess the soil biodiversity, and which are also sensitive to the management or environmental factors, and are also linked with the soil functioning. But for this, what steps do we follow? First, we identify how many indicators are needed to reflect a high percentage of the variation in the community and what is their complementariness. Second, we try to understand if these were reacting or were sensitive to environmental factors and management. And third, the last question is, do they show relationships with the soil functioning? To address the first question on how many indicators are needed to represent a good proportion of the biodiversity, we did two complementary analyses, two complementary studies. In the left part, the left graph, is a dendrogram showing the correlations between each of the metric of diversity that has been measured in this study, where the more intense the blue color are, the more negative associations, and the more positive, the more intense in color, in red, the more positive. And for here, we can show that the more close are also the cells, it means that show a similar response in terms of diversity variation. For this, we identify 4 clear clusters of metrics that relate similarly in the different soils that we assess. And in the second graph, we have a principal component analysis where each of the component, each of the dot, represent the cumulative variation of the total biodiversity, let's say, biodiversity concept in the soils that we have. At first sight, we can see that if we would like to have 100% of information from this soil biodiversity, we will have to measure almost all the metrics that we assess, where the curve reaches the plateau. But for now, we will focus on the properties, on the metrics, sorry, that explain more variation in this whole soil biodiversity. For the first, we identify the first cluster that is based on molecular methods. And within this cluster, the prokaryote richness was the component that explained higher variation in this first component, let's say. Following with the second cluster, that is all related with kind of the PLFA's data, we identified that the fungal abundance within this cluster was the main important metric. That means that explain more variation in this component. third and fourth, we can make sure that we identify the faunal-related data in terms of abundance and richness. And for this, the mites or the collembola abundance were the ones that explain higher variation in this component. And fourth, the total microbial storage biomass and arbuscular mycorrhizal fungi. So at this point, we can answer one of the first questions, that how many biodiversity metrics do we need to have a general understanding of the soil biodiversity? For our study, we can say that with 4 measurements, we can assess the 70% of variation in our data set. Moving to the second question that we have to address is how they are sensitive to the environmental. For this, we perform again a correlation analysis where the more intense the more blue intense color is, the more negative, and the red is more positive, with asterisk in the cells if these are significant. And we can see each of the environmental factors in the left, and in the bottom the different candidates for soil biodiversity indicators. Navigating a bit this plot, we can observe first that there is a general negative association between temperature-related variables and prokaryotes richness, fungal abundance, collembola abundance, and mites abundance also. If we focus also again in the prokaryotes, there is a negative relationship with pH and clay content that is also observed for the mites richness, for the collembola abundance. Focusing a bit on the total organic carbon, that is also an important component of the soil, there was a general positive association between the fungal abundance and the mites abundance with this component. Moreover, looking a bit on the arbuscular mycorrhizal fungi, we can see that these are negatively related with precipitations or also with bulk density. And now at this point, it's important to know that we are talking about soil biodiversity indicators that are sensible to environmental factors, but not necessarily all. We found that were sensitive to soil management. That is important to note at this point. Among them, the total microbial storage biomass and the fungi were the ones more strictly related with management, and the collembola abundance, in this case, the lowest. Moving to the third question, we assess what is this relationship of these candidates with the important soil ecological functions. In this table, we represent the different the correlation values, where the red numbers represent negative associations, the black the positives. In bold, when these are significant, and highlighted in blue is where this metric has the highest value of relationship with the functions that we assess. For this, we first can say that if we focus on the blue highlighted cells, the prokaryotes richness and the fungal abundance, they're the ones that we higher relationship with the functions that we measure. In particular, the fungi with the multifunctionality, and also interesting with the carbon stocks. If we focus on the food regulation or the crop yield, in this case ecosystem stability, that is how much variance they have in terms of naturalness in the surrounding environment, we can see that the prokaryotes richness and meats were negatively related with that. That is interesting at this point. Moreover, moving a bit to the mites, we see that these are positively related with water availability and carbon stocks. And for the total microbial storage biomass, these are positively related with the nutrient cycling in general terms. And it must note here that from the 6 candidates of indicators that we have, the third and fourth point, we have 2 of them that can be really useful to represent a higher percentage of the community. But we identify the mites and the total microbial storage biomass as the third and fourth indicators because of the high relationship with carbon stocks and nutrient cycling, respectively. So at this point, we would like to make a summary of these results. Across all, we have 4 soil biodiversity descriptors that are technically and conceptually complementary between them. And must be note that these are the SOILGUARD indicators that this provide a standardized approach to monitor the soil biodiversity. And this data set of indicators can help to assess the soil biodiversity, the soil health if we understand this as the functions that can be related positively or negatively with them, and also the impact of climate change or management in a cost-effective way. This depends on what is our perspective. If our perspective is how the higher number of metrics that we have to measure from the soil, this one with 4 metrics we can have relevant information from the soil. I will stop here. This is our work that we performed with our data set, and now it's time just to receive feedback from other projects to see what is happening there, and for sure validate with other data sets in a similar format. That's all. Thank you very much. Questions, yeah.
Yes, stay there. There's time for questions. 1 or 2. Aha. The mic will come to you.
Yeah, why not? No.
Yes, my question is about the molecular methods that you were saying. Could it be it's just because we don't have sequence of all organisms? This is a problem, of course. Because you mentioned a lot of criteria for the molecular method that didn't work, but could it be that we don't have a sequence of all microorganisms, right?
Yes, I mean, this I also mentioned that the databases are quite important, and this in particular for microarthropods, those are not well defined yet. But well, for nematodes, we are going quite forward. But yeah, no, definitely the database is quite important limitations.
And about the relic DNA that you say, could it be that it is also horizontal gene transfer? Because, for example, for agrobacterium, we know that some genes are transferred to the plants. Could it be that it's also the case for the network that is in the soil? I mean, maybe it's things that you can search further, just as a suggestion.
So you mean like some genes transfer to some genes from nematodes? No, some genes from bacteria transfer to some.
In the soil, it could be that they get some genes from some other organisms. Maybe this is something that you can also search further. This is just as a suggestion.
But I think it's very difficult to see that from.
But
from
for
the
the
results we have.
agrobacterium to the plant, it was easy, but I don't know. Maybe.
May I add something here? Like this circularity of the genes between organisms is also happening, but with the molecular methods, we amplify specific regions that...
ITS for eukaryotic and 16S for prokaryotic.
Yeah, and at the end, are you assuming that there is a transfer between this gene and the agrobacterium, or because?
No, I don't mean exactly that, because, okay, this ITS and 18S and 16S, these are the very norm things that normally for prokaryote and eukaryote it is using, but no, I mean not that specific genes, but maybe just as a suggestion to put it as a horizontal gene transfer in your set.
Yeah, this is very interesting. Thank you.
Thank you.
For another research project, maybe. Yeah, Mirko.
No, so just for coming back on the last table, because it's important to have this synthetic table, just to understand very well the two-column response for soil organism and change in ecosystem functioning. I assume, in fact, response of the soil organisms is the way the soil is and how the element react. But after the last column change is a positive change. What is the last column? Change in ecosystem functioning. That means we expect to have a positive impact of.
This is terminally basic on the conceptual thing that we assume that the external factors are impacting the biodiversity, and the biodiversity is impacting the ecosystem functioning. And what we found is that there are relationships, but we assume that this is an effect, let's say. but a current effect that was observed with the soils that we have. It's not a prediction, it's this an assumption that they can introduce or could be potentially linked. Maybe the correct word is link, not changing, but the links were found between these metrics and the soil functioning. Thank you.
One at the end, two at the end. The last two at the end.
Thank you. What excellent papers we've had this morning and this afternoon. How are you actually measuring soil compaction?
Soil
And
compaction.
measuring its effects, the effects of it?
Yeah, we use the bulk density to assess the soil compaction. When the higher bulk density we have an assumption that is the higher soil compaction. This was the function that was, yeah.
And I think that's it. Okay.
Okay.
Thank you. And now you can go sit down.
Yeah.
Done.
Yeah, thank you.
Well, we're not over now. We have a panel discussion, and I will call to the stage. I will butcher your names, but I'll start with Thomas, Gerg, Diedrich, and we have Natalia online. Do we? Yes, we do have Natalia online. She informed us this morning that she was sick, but she was still able to join us online. We make them change the names.
Okay.
Okay, is Natalia listening to us? That will be important. But meanwhile, we can present each other, right? Natalia's already online. She comes from John Deere. Thank you. Then we have Tamas. Yes, you are a researcher and also coordinating the Lamasus project. We'll be curious to see the links, but we come to that in a second. Then we have Diedrich. I'm butchering. Tell your own name.
Diedrich de Kelling.
Exactly that. From Agri Land. So it's a land manager at Agri Land, which is an advisory service. Putting it short. And then we have Gert, which also is managing a project, but on regenerative agriculture, if I got it correctly, right? So ReNature.
That's correct.
And we will also see the links with the more theoretical projects here in a second. I don't know who wants to start. Maybe we go with Tamas?
Sure.
And mostly
Sure.
the link between your project and what you heard so far, including this morning too. Because otherwise it's too specific.
Okay, yes. Does this work? Okay, yes.
Now it does.
Okay, no, yes, okay. All right, so thanks a lot. No, this was very interesting, this session in the morning. So Lamasus is also evaluating the impacts on soil. And Lamasus is mainly about the policies that drive land management and which impact on the soils, both on the biodiversity side and also general biodiversity, but also on the climate side. So on the climate sequestration and what we have on the soil organic carbon in the soils. And it is also looking at the farm profitability and the trade-offs thereof. So it looks at the agriculture and the forestry sector and essentially looks at how these land managements are changing. We have a very strong forward-looking component, so I was very happy to see here these deep dives into the different management systems. Especially, we have also management systems in Lamasus which are defined a bit differently, but we do have organic in there. So that was a very, very interesting connection because we would like to assess in a forward-looking manner, also looking at the economics of it, so prices and trade flows across Europe and also across the globe, of what impacts of different policies are. So for the agriculture, it's very strongly the agricultural policy. but we also have environmental, regional policy, and there we look at the medium to long-term impacts. And there we can we would like to see, of course, organic payments are a key aspects of that, and how they would impact essentially the soil biodiversity. We have some biodiversity indicators in there, so we are looking at it with species abundance and the biodiversity index index. We can also take into account different taxa. So that's possible, but of course it would be very interesting to see whether some of the impacts that you see here and some of the indicators are viable, and then whether we can find policies where we incentivize the biodiversity protection of the soils.
Now we move to Dirk on the ReNature project and what your project is also doing with One Soil Biodiversity, and if there's any much links with the one we just heard about.
Yes, thank you. So ReNature is based in Amsterdam, the Netherlands, and we are executing projects mainly in the Global South, where we help the smallholder farmers make the transition possible to regenerative or agroforestry farming systems instead of the conventional ones. We mainly have the approach of having a model farm from 1 to 4 hectares, where we test, where we monitor, where we provide support to the farmers. And then after X amount of years, often around 1 or 2 years, we scale up to several hectares to hundreds of hectares, where we have we provide less and less support, because the whole idea is that the farmers and the community become more independent with these new farming systems. And yeah, this is mainly what ReNature does.
I will ask you about how you measure biodiversity in your project later. Yeah. But before we go to AgriLand, yes, Diedrich?
Yes, AgriLand. So I work for AgriLand. AgriLand is basically an agricultural company within Belgium, where we cultivate approximately between 8,000 and 9,000 hectares in Belgium. And myself, I'm responsible for the Flemish region, basically. So, yeah, that's it. Basically, we are, of course, interested in biodiversity within soil, because we see this has a lot of impact on our economical result at the end. So,
do you see that the members of AgriLand are increasingly more interested in biodiversity, or not yet?
Yes, they are, because with the climatic change we have also in Belgium, soil has to be more resilient, definitely. And I think biodiversity within soil has a great impact on resilient soil within agriculture activities.
Thank you. And last but not least, Natalia, can you hear as well? Maybe not.
Yes, I can hear you. Can you hear me well as well?
Yes, go ahead. Brief introduction.
Okay, thank you, and apologies for my rough voice. I've got a cold and could not join you in person today. Apologies for that. And so, thank you for the invitation. I'm part of the John Deere company, and some of you might have heard about this company, US-based but acting globally, and we are one of those who serve the farmers and the contractors in rural areas to farm their land in the most sustainable way. And how will we help them? Of course, by providing our machinery, agriculture equipment, as well as, even more importantly, precision agriculture solutions. And precision agriculture solutions, you can think of a broad range of digital tools and applications, different old but also new, like really breakthrough technologies that are already on the market partially. Just to name one most recent example from John Deere. It is a spot spraying technology, we call it See and Spray. that really uses like computer vision and AI-powered with a sensing algorithm to scan really on the field crops and distinguish between the crops and the weed. And then the individual nose control activates only those nozzles on the boom of the sprayer where the weed has been detected, and only the weed is being treated by herbicide. And of course, this is really nice because in that way you can save a huge amount of herbicide like glyphosate and all these bad types of things. So you can potentially go with savings up to 70% already on some fields, and even more is possible because the AI-powered algorithm is learning constantly. The better data it gets, the better decision it can make to help farmers really to control weeds in the most sustainable way. And of course, it also tells a story about how it contributes to the biodiversity, to preserving, to enhancing biodiversity, both on the field, like below the soil coverage, but also below the soil ground in terms of soil biodiversity. This is just one example, but of course we are working together with farmers, together with other industries, with research and educational institutions, to really understand the impact of different farming practices, of regenerative farming practices, for example, like reduce soil disturbance in terms of reduced tillage, cover crops introduction, enhanced crop rotations, that really have very practical input on the soil health outcome, because really we believe that functionable and resilient soils are the key for the future-oriented, sustainable, competitive and resilient agriculture. And just to give you one practical example, which might be more related to the conversation of today's event, is we are working right now in US with the Soil Health Institute to develop what we would call a soil health dashboard. And this really could make this trial to link the impact of farming practices, of more sustainable farming practices, on the state of soil, on the soil health and soil biodiversity. And then we would look, for example, into the indicators like carbon, soil organic carbon, carbon releases dynamics, or also soil aggregate stability. So some of these indicators have been already mentioned before by other speakers. Maybe I'll stop here. This was just in a nutshell to give an impression where we stand and what we are eager to do together with you guys. Thank you.
Thank you, Natalia. Yes, you mentioned a lot of sustainable farming practices, and there is this this balance between should we promote and support farmers on the management practices per se, or on result indicators. And in this case, indicators, you have the most common ones like organic matter, and more increasingly biodiversity. How do you see in your projects and also industry how the balance is done? Are you still more on the list of management practices or more result indicators? And on result indicators, have you already gone as far as looking into the biodiversity? Maybe not with what we heard today, even potentially tomorrow, but have you thought about this dichotomy and this balance? Who wants to start? Go, Thomas?
Yes.
Okay.
Yes, no, that's an excellent question, and I think for Lamasus it's also very topical, because indeed we are now, next year is the last year of the project, so we are developing our scenarios that we will run, and there indeed we are playing through multiple policy pathways. So we will look towards something which is more similar than how the agricultural policy is set up now, which rewards in the sense the practices, and we have payments for practices, I mean, with some restructuring potential towards the future and some of the simplification that is envisioned, but more or less in a similar vein. And we are also looking into scenarios where through our models we would like to see how more results-based payments are done. And this is currently, we were looking there mainly through soil indicators, so soil organic carbon buildup and how much is stored there. we can also link to a certain degree the management, but I think the soil organic carbon, we had it a bit more detailed, our analysis. The biodiversity side, it was a bit more complicated to assess this. So I think currently we are looking towards the soil organic carbon, and we can assess, we can at least give a boundary to what such a rewards-based system could look like, and what we could achieve as at least in a minimum condition with this, because of course it's not very easy to update our models towards a very different payment schemes, but I think this is something that we are very excited to try out a bit more of a results-based methods, and where this could land us in terms of biodiversity outcomes across Europe, in terms of environmental outcomes, and what this would mean also for farmers' income and for equities across small and large farms, etc.
Thank you.
Yes.
Yes.
So for our project in Ghana, where we monitor the impact of agroforestry for cacao production, we see that if we use management practices such as less tillage, but also using cover crops, shadowing, just in general diversifying the farming plot, it also enhances soil biodiversity. So that's really on the management side, that we already have seen a proof of concept within our projects, not only with cocoa, but also with coffee or other commodities, that diversifying the plot also enhances soil biodiversity. But there's also experts involved from Ghana that already have experience with agroforestry in cocoa, that already showcase what the impact could be. So that's more backtracking, looking at the results that already have been monitored in the last 15 years, and then seeing, okay, this is what we can promise with a certain range of end result, but we can promise, okay, the soil organic carbon or the soil organic matter, the soil biodiversity in general, we can reach it to this and this level because we have these experts in our network and in this project. So.
Yes.
At our level, let's say, the main parameter that we measure within our soils is the total organic carbon content within our soils, and that's basically the main parameter that we follow up. We try in general to increase this parameter because we see that it has a lot of effects on, again, resilience of the soil against erosion and other very important parameters. So the nice presentation that we had just before, that's kind of parameters that at this point we don't measure, clearly not. Why? Because I think we don't have the certified or accredited laboratories to measure these kind of things. But of course, yeah, I think we are interested to go further than the total organic carbon parameter, and if we have some other interesting parameters to follow up our soils, of course, we are interested in that. Yeah, definitely.
Natalia, you have something to add at this stage?
Not really. I would just echo the comments from the colleagues, and I would also like to underline that most of the parameters we are looking right now, they are not talking that much yet about real biodiversity, like what is really happening below the soil coverage. And for very simple reason, if you want it really to be on large scale and be implemented like at every field and at every stable and rural areas, it could bring complexity and cost. And at the end of the day, it's the question, should be the farmers in charge of like applying this technology on the spot, or how should it be measured? Like what is the right balance between like laboratories measurement and on field, on spot approach in the future? Where do we want to go with that? And if the farmers need to be a part of this journey, they will also need a kind of a practical and affordable solution for that.
You're opening another question on who owns the data. Do you have comments on that, on this? Another balance to be made between the farmer that indeed should own the data, but also industry and policymakers. Any comments on this one?
Yes, data, that's indeed a sensible topic for sure. We've got, for example, indeed propositions from commercial companies to scan our fields, scan our lands, and give some data on how general health of the soil is. And of course, when we've got these propositions, we are a bit careful. Personally, I would prefer to have some data analysis of soil information from official universities or laboratories, and not at the first place from commercial companies, because we think there is maybe a conflict of interest in these kind of propositions. So yeah.
That was clear. Anyone else wants to comment on this one? Natalie can also respond. No?
No, I completely agree to work with you.
With whom? I assumed, just to clarify.
Yes.
So I'm assuming because most of you focus on organic matter. Not surprising, there's much more history behind. We have is well known, right? There's international standards, which don't happen yet with biodiversity. But do you still see? Do you see that? Well, tomorrow or the day after tomorrow, that this, if this is a thing, do you have a practical view on how this could actually help in a day-to-day practice, or in terms of yields, in terms of, because we're talking about biodiversity, and everyone agrees that it's a good thing. But for instance, to sell to a farmer, do you have the practical arguments to do so? What you require from research and also from the indicators, apart, imagining if they are standardized and if they're cost effective, would you be able to sell easily to your farmers to have this already put in practice? Was I clear? Maybe not.
I think farmers can react positively to this kind of propositions when they really have problems on their land. Then you see farmers think about their practices and think about alternatives to resolve the problem, the practical problem they have on their land. And that's practically what I see on the field. As long as the yields are almost correct, they don't feel really a need to change something until problems start to appear. Yeah? And that's I think in practice most of the time the trigger to start thinking for alternative practices. Yeah.
So it'll be important to make a more clear link between the benefits of biodiversity or specific type of biodiversity with management practices. A bit like we know with organic matter that it has a more resilience for climate, as we saw this morning. So a similar type of. I think I'm leading our panelists into certain conclusions, but I'm happy that you agree. On the biodiversity indicators, we're talking generally. Do you think that there's what else is missing? If you want research to focus on something, apart from this argument on make it practically, a more practical argument, is there anything else missing that you would require, that would want research to focus on?
I would say, in general, don't focus on too many parameters, because if you're working with farmers in the global south, the monitoring of soil biodiversity needs to be applicable, suitable, accessible. It needs to be financed with very low investments for the farmers. So it needs to be practical with simple pH stripes, for example, or visual soil assessment before we starting to dive into the labs. It needs to be it needs also to be easy to be executed with the farmers. And that starts with showcasing what the benefits are from biodiversity in soil, comparing that with status quo, so with conventional farming.
Because at the moment, are you measuring biodiversity in your project at all? And how?
Yeah, so we're doing both. And we're doing both for several reasons, but the main reason that we're also doing the more costly lab assessments or with the universities is because we need to showcase also with data that what we are doing is suitable and good for long-term vision. But we are also focusing with capacity building training on how to monitor with visual assessments that are already very useful, where we can understand better what water retention, soil structure, and all different kind of indicators are, and it doesn't cost anything. So that's way better for a scalable, for scaling projects and showcasing what the benefits are of soil biodiversity or agroforestry.
Especially if you work with the Global South, right? Because
yeah, especially for
labs availability and expense might be different.
Yeah,
differ
but
a little bit.
a lot of crops come from the Global South, so yeah, it's a big market.
I will open up to the audience for questions. Haha, and we have already a winner. Please present yourself. Yes.
Hello. Sorry, I'm Caroline de Meus. I'm a farmer in Belgium. We changed our practice 8 years ago, and I'm quite surprised that you say farmers are not able to determine the biodiversity of their soil, which is not true, and it's not so complicated. Of course, we will not count nematodes and etc., but we will count soil worms, which is very easy. And if you don't want to count them, you can at least see their houses. I don't know how you call that in English, but it's very easy to see on your land. And you have to look at the structure of your soil, which is very easy. If it's compacted, water retention, the roots, you can see how deep the roots go. So there are many other ways than to analyze biodiversity in the soil.
Is any of you doing that exercise? And if you're not, why not?
Yeah, well, yeah, you're absolutely right, madam. And in some occasions, we go further than just the total organic carbon on our soil analysis, but we don't do it systematically. And we think we go further when we see some problems within the land. That's again, mostly the trigger to go further. Yeah?
But to talk with your farmers, you can start with very easy things. You don't need to be very complicated or very scientific to let them know what to look at or what to do to have a real change in their soil.
Mm-hmm.
Thank you.
Any other question?
Thank you very much. Very interesting, thank you, panel. The triggers that you see, which are the practical problems which focus the farmers to actually perhaps make changes, what are they, please? And a second question to that is that very often soil degradation further away from the problem occurs. So you have a lot of severe flooding downstream, and that's a big wake-up call to the guy who suddenly finds, like in Lincolnshire recently, that his crops are completely submerged for months on end. And are these ideas for improving soil health spreading beyond the territory that they affect?
Well, yes, I can speak for the conditions we have here in Belgium, but indeed erosion is most of the time the big problem when farmers start to wake up and when they see erosion problems. We have some erosion problems on some of our fields, and it's a big problem for sure. We don't want to see these kind of things, and then to try to go back to a better status of these kind of soils, it's difficult and it takes a lot of time. So erosion for sure, I think that's the in these conditions in Belgium, I think the first trigger to the first wake-up call for farmers, yes, I think.
Maybe I wanted to address on the uptake of the practices that you initially mentioned, right? And there, I mean, we looked at a fair bit of indicators of when farmers adopt practices and when not. And I mean, there is of course economic aspects of it, and farmer age matters. And I think what was fairly robust is that kind of what your neighbors are doing is very important. So this kind of farmer cooperatives and organizations are super important. That was a really surprising result that through multiple studies with different methods emerged quite clearly. So I think that was quite interesting on where you can kind of invest.
So knowledge sharing. Gerk, you have also something to add on this in your project. Are you also looking at individual assessment that you mentioned? Is earthworms also something that you consider?
Yes, that's also one of the I don't know who questioned it, but yes, earthworm counting is one of the assessment to cut. Yeah, it's very common practice to know what soil biodiversity is, because worms tell a lot about how healthy the soil is. Yeah.
Is there any other question? Yes, there.
Hi, Katie McRobert from the Australian Farm Institute. Looking at the title of the panel, which is about harmonisation and some of the challenges around harmonising measurements of soil biodiversity, I think even within this panel, it was clear that there are some differences in who you would trust, who you'd see as a trusted advisor or a trusted arbiter of a standard. Could I ask the panel to give advice? You mentioned that soil organic carbon was one thing you measure, partly because you can, but also because there is a genuine recognised standard for that. Who should set a standard for soil biodiversity? Where do you place your trust for that standard setting?
Aha, who wants to start?
Well, as I said previously, I think we trust more official companies or labs before commercial propositions from commercial companies. If these labs, official labs, are certified, of course, that's a very important point. Yeah, that's what I think about it.
Yes, I mean, maybe also from our side, I mean, we mainly, so in Lamasus, we do not have any kind of first-hand collection of data in that sense. So we use public or data sets or data sets that have been officially collected or that are available. So I think those, from a scientific point of view, are always preferred to private enterprises because it's just much easier to get. So that's based on that. Of course, there has to be some scientific consensus, I suppose, on what the measure is, which is, I guess, at the crux of the matter, because there is, especially in biodiversity, there is lots of indicators available. So that's will always be a challenging one to arrive at.
So you say that the standardization is the key on this aspect. Natalie, you want to, because I don't look at you, I feel that I'm ignoring you a little bit. I don't know if you want to say anything.
All is good, and I hope you still can hear me well. I would like also, again, to continue this provocative discussion of what would be actually the long-term vision for soil biodiversity. Are we here talking about keeping this a kind of silo with labs which would measure maybe more precisely certain parameters on biodiversity, biodiversity, but it would be maybe not in real time and with less samples as compared to potential foreseeable future technology on spot on the farm, which would be kind of easy to use and accessible to an average farmer, because it's actually up to the practitioner, to the landowner, to the operator on the land to really conduct the farming practices, change the practices towards more sustainable and regenerative agriculture, and then to see the outcomes. So being able really to connect the dots between what do I need, how do I need to change it, and what do I get at the end of the day. Because thinking about all the three dimensions like economic sustainability, environmental health, but also the social sustainability in rural areas. So how do we get there? I think so the next panel will for sure address some of these questions, because they relate also to the forward-looking policies in Europe related to agriculture and supporting enhancing soil health and other indicators of sustainable agriculture. But this way or another way, I guess every technology is kind of complex and comes at certain cost. And this cost must be affordable, solutions must be practical, and farmers would always need a kind of advice, like where to start, like do I need that, like okay, I know I'm facing a certain problem, like maybe I'm facing increased erosion or dryness on my fields, or I don't know, something that is a kind of a big problem, farmer has recognized that, so what does he or she do about that? And here we must also talk about kind of advisory systems, training and knowledge support for the farmers, that they are kind of equipped with the knowledge, and then they can decide what they do, how they do they go for the like certified laboratories solutions, or do they go for independent private companies if they would offer any of practical solutions? They could ask their neighbors, they could ask industry advisors, they could ask their trusted dealers from different input supply companies. So I guess this entire area of extension, training, knowledge support, and then sharing the research project results, bringing that into a large scale to the farmers community is a piece that should not be forgotten, in my opinion. So farmers should not be forgotten on that journey.
Thank you. Any further question? Yes, Charles. Kind of blind.
I come from the west of the Atlantic region. Our problem is how to maximize our yields and reduce our costs. I've handed over to my son, who's now looking at all these things, and what I think we need is a template of the analysis we should give, simple and practical, that we can feed to the scientists to get practical advice as to what we should do. And I think that's what we need. So it's not really a question, I'm afraid.
It's a comment.
It's a statement.
It's a comment that I'm assuming everyone agrees. Yes. So I think I maybe can try to summarize briefly. Well, at least what was the consensus? Everyone seems to agree on the importance of having biodiversity indicators. It also seemed to me that currently the focus is the ones that are already proven, standardized, and relatively cost-effective to monitor. Otherwise, the reliance is more on visual, and that works well for biodiversity, for fauna when it comes to biodiversity, but not so much to the world that we don't know much about when we talk about the other type of soil biodiversity. Nonetheless, it seems to me that everyone agrees that we should focus more on how to make it standard, how to make it more practical, how to make it more easy to use, how to help connecting the dots, also to give the arguments, and also to give the tools for farm advisory services, and also for projects, and also for industry, to give options to farmers to develop more soil biodiversity. Was I clear? Well, I hope so. Nonetheless, that's not a problem, because we will have another panel now still focusing on soil biodiversity indicators, but also now with a more policy focus on how to make this link. And I wish them luck. So let's give an applause to our panel. Done.
Thank you. Thanks a lot.
Oh, I should announce the coffee break. Well, there's a coffee break.
I also get hey, nice to meet you. Hey, nice to meet you. So it's a bit tight. Yeah, here it could be glamorous.